we have identified a group of proteins that they are binding to the repeat regions in healthy subjects. This group of protein is called polycomb group of protein. The only thing that you need to know that is relevant about this protein is the fact that other people before us had shown that these proteins are associated with binding to a region. What they do, they bind to a region, they make this region compact, close, inaccessible to other proteins, and in doing so, they shut off expression from this region. So if a polycomb protein binds to a region is making it very compact, other protein cannot get there, and so that's the way they cause absent or low protein production from a particular genetic region. We found that this is going on in healthy subject at the level of the locus responsible for the disease. And what is actually going on in FSH patients is that the deletions reducing the number of these D4Z4 repeat that you all know are involved in FSH is, so when you reduce the number of repeat, you also reduce the amount of these polycomb proteins that are present in the region. So because they are present in a reduced amount, they are no longer capable to efficiently silence, to efficiently close, maintain the region in a closed conformation, and so the region opens up. And these allow the access of other proteins to the region that actually are responsible for activating the protein production from the region. We actually, because in FSH nothing is simple, we actually found out that the process is not going exactly like this, so directly from losing of the repression to activation of protein production in the region. We found an additional player in this process, which is a so-called non-protein coding RNA. So I don't know how much you are familiar with the way the production of protein is going on in our body, but basically the way a protein must be is written in our DNA, and the information must flow from the DNA to the machinery that makes proteins. The way this is done is because the DNA is written, and this is used to make what is called a messenger RNA, which is a little RNA that contain the information that is read to make the actual protein. For many years, this is the only form of RNA that it was known to be present in the cells. So the flow, the information in the cells was considered to be just this. So you have DNA, it is read to make messenger RNA. Messenger RNA goes where the protein building machine is present and is read to make proteins. More recently, we have discovered that there are new classes of RNA which are actually not used to make proteins. They are used to regulate the production of these RNA that make proteins. And so we have found out that there is one of these new form, non-coding RNA, that is produced from the FSH locus, and this is absolutely required for the increased protein production from the locus. So you need to have this non-coding RNA to be made in order to have the downstream effect to take place. And we know that because there are tricks that we can use in the lab. We can remove this non-coding RNA from the cells, and if we do so, we don't see any of the downstream increased protein production from the FSH locus, which in a way is a very interesting finding because it suggests that this non-protein coding RNA can be a therapeutic target, because in our end it is absolutely required for the switch that lead to the increased production of proteins that you have read in the literature, like DAX4 and other proteins from the locus. If you takes off the non-coding RNA, there is no increased production of DAX4. So obviously, one of the line of investigation that is going on in the lab is try to study how we can control the production of this non-coding RNA in order to, we hope, normalize protein production from the FSH locus. Now, another of the things that we are also doing is to understand how the increased production for the different proteins that are present in the region may be responsible for the disease. Again, you may be familiar with that during the years, the field have experienced a lot of hype for a particular protein that was at that particular time the key target for the disease. We should go and look for this protein, and that will be the end of the story, which was followed by a down, where there were less enthusiasm for that particular protein, and afterward that was followed by enthusiasm for a new protein. So for people like me that has been involved with FSHD since, as I said, quite a few years, I have experienced these up and downs quite a few times, which is very frustrating in a way. And so you may know that there have been several potential candidate genes that different individuals have proposed for be relevant for the disease. In particular, there are listed the 4 genes that at various levels different groups have put out as possible possibly involved in the disease. And so the important thing is that one of the ways you can try to understand if a particular gene, a particular protein may be directly involved in a disease is to generate mice that display exactly the same feature that has been found in the patient. So if in FSHD patients you find increased expression of, let's say, ANT1 or FRG1 or DAX4, you can make mice that express high level of that particular protein and see if they develop a muscular dystrophy. So this has been done by me when I still was in US and by other groups. And out of this work, the only gene for which there is a clear indication that when it is overexpressed in a mouse lead to muscular dystrophy, this is a gene called FRG1. This is the only one that when by itself is overexpressed in an animal, allow the animal to develop a muscular dystrophy. And actually it is a muscular dystrophy that displays several interesting features that are conserved in FSHD patients. Now there may be technical reasons why, in particular for the case of DAX4, the work that has been done has not been successful. And there are better ways probably that the people have to follow in order to develop new animals overexpressing DAX4. But at the moment, the only animal model that displays some of the FSHD feature is the FRG1 mouse. So we are actually then mainly concentrating on this model because it's the only one available, and we are doing so by investigating both how FRG1 overexpression may lead to muscular dystrophy, but also we are using this model to test possible therapeutic approaches. So again, there are a number of ways that you can think that you can use to develop a therapy for FSHD. You can go directly at the source of the issue, and as I said, you can develop way to control, block the excessive production from the proteins of the FSHD locus, and we have a way to do so, and I will show you an example of what we can do. But the other thing that you can also consider doing is go a little bit more downstream. So instead of addressing directly the source of the issue, address the consequence. So once you identify the way FRG1 or DAX4 work and which are the way through which they cause the disease, you may think about developing approaches for affecting this downstream effect, not directly the source of it. And we're moving in all these directions. So as an example of what we can do in order to directly address the source of the issue, so again, the issue here is that you have one or more proteins that are produced at higher than normal levels in FSHD patients. So the most obvious approach is then go there and eliminate this protein. If you have too much of this protein and if this is the cause of the disease, you can simply eliminate this excess of this protein, and this in theory should translate in an amelioration of the disease.